This topic lecture covers one of the three main structures of the cytoskeleton, the intermediate filaments. This is one of four topics we will cover as a part of Chapter 17. Here are the objectives for this topic. As always, please let me know if you have any questions on these or need help working on one or more of these objectives. This should be a brief topic lecture. In this lecture, we are going to talk about what intermediate filaments are, what they do, and how they work in concert with the cytoskeleton. Before we dive into the three main filaments that comprise the cytoskeleton, let's get a refresher on what the cytoskeleton does for the cell. It is easy to think of the cytoskeleton as the bones of the cell, and that these are hard, immovable objects, like our bones. But that isn't quite true. These filaments are more of a combination of muscle and bone. They provide support, but they are also flexible, as you will see in the next few topic lectures. There are four main functions of the cytoskeleton. These are provide shape, organization of interior components such as organelles, interacting with the environment, and allowing for movement. Cell movement can be as dynamic as cell crawling or as routine as cell division. So what are intermediate filaments? These are rope-like structures that participate in the cytoskeleton and can be found throughout the cell. These fibers are named intermediate filaments because they were first isolated in muscle cells and are larger than actin fibers, but smaller than myosin fibers. These structures all begin with an alpha helix-shaped amino acid sequence with a globular head and tail at each end. While the middle section of the protein is conserved between intermediate filaments, the heads and tails of these proteins vary between types of filaments. These monomers will join together to form a coiled coil dimer. More dimers are then recruited until a series of tetramers are formed. These tetramers are twisted into a 10 nanometer rope-like fiber. There are two types of microfilaments broken into four classes. These are the keratins, vimentin and vimentin-related, neurofilaments, and nuclear lamins. Three of the four classes of intermediate filaments are found in the cytoplasm of the cell, specifically in areas that are subject to mechanical stress, where they can provide reinforcement to the structure. Let's evaluate each of these classes a bit more. Keratin is the most diverse of the intermediate filaments. This is found in all epithelial cells and allows for them to be flexible and help them move with the body. Vimentin is found in connective tissues and helps anchor organelles within the cell and also provides a level of flexibility to the cell. As we talked about in the earlier slide, intermediate filaments reinforce the cells. In this case, the actin of nerves can be very delicate, and these intermediate filaments help reinforce the axon of the cell. The last group is the nuclear lamins, which reinforce the nuclear membrane and help provide support through a meshwork of intermediate filaments. Let's wrap up this topic by reviewing the role of intermediate filaments within the cell. As we have discussed, these rope-like filaments serve as reinforcement in the cell and anchors organelles. They also participate in cell-to-cell junctions known as desmosomes. The big take-home message for intermediate filaments is that they help the cell stay flexible through reinforcement. This is the end of the topic. Please review the objectives for this topic and let me know if you have any questions.